This video will walk you through a real example of using a neural network in practice. More specifically, we're going to use what's called a convolutional neural network, or a CNN. But a bit of a disclaimer before we get started. In this video, I'll briefly explain what a CNN is when we get to the modeling section of this notebook. But we haven't covered different types of neural networks in my channel yet. A more in-depth video explaining a CNN will be coming later, but for today, I wanted to focus more on just using a neural network in practice. This is a short project that I created and ran for the purposes of this video only, so by no means is what I'm going to be showing you today the most perfect model or anything like that. But this video should be a good walkthrough of an actual coding project that uses a neural network model for classification of, in this case, images. Now, even though this is a code walkthrough, and I'm going to be showing you my code, I won't be explaining the code line by line. I think the benefit of this video should be the overall flow of what a machine learning project is like, so that you can try something similar on your own. So I ran the notebook already, and I'm mostly going to be explaining the process I followed, rather than explaining the code. In fact, I used an AI to help me with a lot of the code, and you should do that too. There's no shame in using something like ChatGPT to help you. Just make sure that you understand what you're doing and don't overly rely on them. So without further ado, let's get started. As with any project, the first thing that you will see is setup. We install and import the packages and the libraries that we're going to use. The data that we're going to be using is going to be a dataset I found in Kaggle. This data is called the Annotated Ultrasound Liver Images dataset, which comes with 735 images of ultrasounds of livers. The data comes with the images along with annotations on where the liver is and where, if any, mass regions are present. And of course, these images also come with a label in the form of whether the livers are normal, benign, or malignant. If you want to take a look at this page, I'll have the link to this dataset in the descriptions below. So going back to the notebook, we import this dataset directly from Kaggle itself. We can do this using Kaggle Hub so that you won't actually have to download the files themselves in your own computer. And if someone else runs the file, they also won't need to figure out where they got the data from. So it's nice when these kinds of download options are available. We can see that when we import the dataset, it comes in a specific format. With any data sources you download, it's important to know how they are structured. I drew how this dataset in particular is structured. You can see a base directory of 7272660. Don't ask me why it's named like this. But inside there's subdirectories for each of these classes. And under each of them there's a directory where you can find the images in JPEG format and the annotations of liver, mass, and the outline in a JSON format. Now, I'm not an expert, but I take it to mean that outline refers to the entire ultrasound area, that liver refers to the actual liver itself, and that mass refers to the area that has some kind of a tumor or lesion or whatever that you can detect that has some kind of abnormality. And the JSON files represent the pixel areas of the edges of where these areas are in the images. It can actually be kind of difficult to tell just with the data, especially for image datasets. However, we can validate this by just looking at an example. So in this code, I load up the very first image and its corresponding segmentations in the benign category and draw the areas that are defined by the segmentations. This gives us an overlaid image in which we can verify that my assumption seems to be true. You can see a red outline that represents the segmentations in the outline JSON, a blue outline that represents the liver segmentation, and the green outline that represents the mass segmentation. The bottom line is, it's always important that you understand exactly what the data is giving you. In the next cell, I look at 5 random images under each category just to get a feel for what each type of liver looks like. Just from my uneducated human eye though, it's pretty difficult to distinguish between livers in different categories. But let's see what the model can do. But before we get there, we have some EDA to do. First, we already know from the data description that the number of images for each category doesn't match. We can visualize this with a bit of code, and we can see that there are only 100 normal images. I think that's too little for the model to learn what a healthy liver looks like. We can alleviate this issue a little bit by a process called data augmentation, which we will get to in a minute. But before that, let's also take a look at the sizes of these images. I've plotted histograms of the pixel sizes of each of these images, and you can tell that they vary by quite a bit, ranging from 400 pixels to 1400 pixels. It's useful when the model learns images of the same size, so we can also standardize them. So let's address these two things. First, we'll resize the images so that each of them are equal squares of 512 by 512 pixels. That's what I do in this cell, where I define the target size and loop through the images to resize them. I do this by rescaling the height and the width of the images to the same size for each image and then copy them into another directory so I can have both the resized versions and their originals if I need to go back to them. Importantly, the coordinates of the segmentations of liver, mass, and outline also have to be scaled along with the images. You can do this simply by multiplying them with the same vector that was used to scale the images. Reusing the same code that we used to plot out the first image, we can see what the 512 by 512 image looks like and verify whether the overlaid segmentation outlines also got adjusted along with the resized images. And then we can also reuse the next code to plot out 5 images for each class and confirm that all images have been resized correctly. Now let's address the sample size issue. We have 200 benign livers, 435 malignant livers, and 100 normal livers, and we want to standardize them so that the model learns from a better mixture of them. There's two things we can do. We can undersample the malignant liver images by cutting them, or oversample the other ones by increasing their number. There's pros and cons of both. Obviously, if we reduce the sample, we're getting rid of valuable data. And if we increase the sample, the only way we can do it without our limited resources is to augment the data by copying the existing images, applying some transformations to them, and then treating them as new images. The challenge we face with this approach is that if not done carefully, it can lead to overfitting, since we're essentially training on the same image. So let's combine both strategies for a good middle ground. For this example, I think that the most important thing in a medical image detection is to first and foremost detect when there is malicious mass in the liver. So let's split 50% malicious and 50% benign and normal. So we'll augment 50 benign images, 50 normal images, and reduce malignant images by 35. This should give us 400 good livers and 400 malicious livers. This can be done with the augmentations library, which lets you pipeline the augmentation process with predefined functions that can do these image transformations. This makes augmentation really easy. So I apply these 3 strategies randomly to some images and then loop through both the images and the segmentations to apply the transformations. The majority of this code is actually to apply equal transformations to the segmentation coordinates, which unfortunately is a little complex. The segmentation coordinates representing different areas in the images are something very unique to this dataset in particular, and since the model not only uses the images to learn, but also these segmentation outlines, it's very important that we apply the same transformations to these coordinates. Anyways, the main loop first randomly cuts the malignant images so that only 400 is kept, and then applies the augmentations pipeline to benign and normal images until it reaches the target counts, and also applies equal transformations to the segmentation coordinates. We can check that this resulted in the following image counts, which is exactly what we wanted. Again, it's a good idea to visualize everything that we're doing, especially with image data. This next code plots out the resized images and the resized plus transformed images so that we can compare the before and after and verify that the right transformations happened. Comparing them, we first see that the images that were not transformed were copied correctly. These first 3 sets of images should look exactly the same, and they do. For the augmented ones, we see that either the images were flipped, slightly zoomed in or out, or that the brightness or contrast has changed. We can see that the first augmented image was flipped and looks like it's a bit lighter than the one on the left. We can also see the overlaid outlines of the liver and mass areas were also flipped, which is exactly what we want. And in the bottom example, we can definitely see that the image is darker than the one on the left. So by now we're feeling pretty good with the data and can move on to the modeling stage. In this example, we're going to try 2 methods. First is going to be a binary classification, just to detect if the image has a malicious mass or it does not. So we set up a pandas data frame with the images and their segmentations, and then append a column that represents their labels, 1 if malicious, 0 otherwise. This is what the model is going to learn based on. And then we split them 80% into train and the remaining 20% into test. In the process, we also stratify based on the labels so that the train and test is split with equal proportion of malicious and non-malicious liver images. The next cell you can see is commented out. That's because I have this exact same code in a Python file as a class. The liver binary dataset class contains objects that basically turns the pandas data frame into an actual PyTorch modelable dataset. This just means that the images and the polygons in the images are turned into numeric representations required for a machine learning model. These models don't actually scan the images themselves, but rather they turn each pixel into a numeric value that uniquely represents the pixel. In these next couple of cells, call the new class I defined and apply this process to our train and test. To understand how the model will scan and learn from the numerical representations of the images, we have to understand what a convolutional neural network is. A CNN is a type of a neural network that is commonly used in image detection. Simply, a convolution is a concept used in image processing that works by sliding a matrix called a convolutional filter across the numerical representations of the image. This sliding matrix, depending on the values of the filter, can learn patterns in the image, and multiple filters can work together to essentially summarize the image while reducing the dimensions of the matrix that represents the image. You can imagine if we keep reducing the dimensions of the image, combining them with other techniques such as max or average pooling, and activation functions like ReLU or softmax, we can eventually reduce this down to just one number. And that one number would be the network's guess at the label, 1 or 0. That's a very simplified way to explain what a CNN is, and if you don't get it, don't worry too much about it now. A separate video where I spend the whole time just explaining a CNN is coming. The important thing for today's video is that a neural network can be designed such that you can input an image and it outputs what it thinks the label should be. So let's go back to our notebook. I will build a neural network model with 4 hidden layers. Only this time, the hidden layers are called convolutional blocks. You can design these blocks however you want to, but the general idea again is that you want to reduce the image down to a single output. For each of the 4 blocks, I follow the same sequence. A conv2d is the mechanism that allows the sliding of the filters across the image, and I do that first with 6 in-channels, or feature maps. The reason we choose 6 is because the image, when we turned it into a PyTorch dataset, we turned the images into an RGB image, which actually needs 3 separate feature maps for each color. On top of that, we had overlays in the outline, liver, and mass as distinct features of the image, so we start off with a total of 6 feature maps. The second parameter is the out channel, which is how many output feature maps we want to produce in this convolution. The more we have, the more patterns it will learn, so we want to increase this number throughout the network. But of course, having more out channels means more compute and potential overfitting, so you will need to choose wisely. The conv2d is followed by batch normalization of the feature maps, and then we want to apply a nonlinear activation in ReLU so that the network can learn complex patterns. The dropout 2D is there to reduce overfitting by randomly throwing away a percentage, in this case 20%, of the feature maps during training. This encourages the model to not over-rely on a particular feature. Finally, max pooling will introduce new filters that slide across the feature map and choose the max value from the feature map and creates a smaller version of the map that only focuses on where the largest response was for each filter, effectively reducing the dimensionality. You can see how the dimensions change in the comments of this code. After the convolutions, the final bit is the part that does the classification in a fully connected layer, just like how it would be done in a typical neural network. Because this is binary classification, we use the binary cross-entropy loss, or in my example, a variation of it called the binary cross-entropy with Logit's loss. The optimizer I use here is AtomW, which is also a variation of the Atom optimizer that introduces weight decay that can potentially reduce overfitting. With this, we loop through 5 epochs and train the neural network. The code here is a bit complicated, but honestly most of it is due to the fact that there's multiple dimensions to consider in our example because the model has to learn not only the images but their outline, liver, and mass outlines as well. Anyway, we see that the training loss is reduced through the epochs, which is a good sign that the model is learning. We see a similar pattern with test loss, which tells us that overfitting was not an issue. Interestingly, the test accuracy, which is just a measure I implemented that counts the correctly predicted labels over the total, was reduced but only after the first epoch. If you want to improve this, there's several strategies we can try. We can try to change the hyperparameters in our model, change how accuracy is measured, introduce more samples so the model learns more diverse examples, and etc. But we won't go there for today's video. But instead, I want to try another model. We initially used a binary classification, good liver and bad liver. Well, we have 3 classes, normal, benign, and malignant, so we can build a model with multi-class classification. To do this, we can go back to where we made the PyTorch dataset and redesign our modeling approach. Instead of creating binary labels, we can keep the original labels. Then we do another train test split, and then change the dataset so that it's viable for a PyTorch image training model. Let's try to use the same model, but this time have the CNN model output 3 separate probabilities, one for each class. The model determines the one with the highest probability to its final prediction. When we train, we feed this into cross-entropy loss instead of the binary cross-entropy loss, which should allow us to train in multi-class classification. Only for this, the accuracy isn't nearly as good, and the model is not really even improving. This is probably due to the fact that we just haven't really structured the model architecture with a multi-class label in mind. But we can also blame the lack of data. Remember, we only have 150 normal images, which is not really that much to learn from. In the end, I would probably only stick to binary classification here rather than trying to build a good multi-class model. With that, I will end this code walkthrough here. Hopefully this was helpful, but let me know in the comments if you have any questions. I know I didn't explain all the details of a convolutional neural net, but that'll be coming later, so stick around for more machine learning content.